I'm hoping that the next group, the panel will come up. That's Sam Barnes, Andre Der-Avakian, and Christine Smith. But I get to introduce Vince, who very recently did for me a clinical favor in some data that I needed pulled, and he made sure it happened. So I'm very grateful to you. You are a wonderful clinician. You joined the department in 1999. You're a professor here, and he is the director of the UCSD Neuropsychological Associates and a staff psychiatrist at the VA. And Vince knows, understands, and has his own area of research and has made additional research very possible because of his efforts. So, Vince, thank you.
So thank you, Mark, very much for asking me to participate in this symposium and in this particular group. I'm going to give some just kind of broad observations that I made during the the talks. And then I'll turn it over, or I'll introduce and then turn it over to the panel to take it further. So these were some of the main things that were going through my head, other than what's for lunch, while I was listening to the speakers. And Larry's work, enough can't be said about the work that Larry has done. I recall being a graduate student in the joint doctoral program and him coming to lecture and really realizing how much I was very interested in the study of memory. I think one of the things that struck me that we don't necessarily talk a lot about is this implicit type of learning or unconscious learning and the importance of that. As a clinician, I'm very struck by how we don't have measures of that, yet we realize how important that is in our day-to-day functioning. We don't have any way of really measuring in the clinic how someone learns something at an unconscious level, and I think that's an important take-home message. If you saw on Larry's slide where he talked about the non-declarative stuff, we're now starting to use those concepts to explore a number of different areas in psychiatric illness. Fear conditioning, for example, how is that related to PTSD and so forth? And I think that an important concept is how those things can be unlearned. And I'll talk a little bit more about that. Another thing that struck me too is just this idea on one of the slides where Larry showed that initially the hippocampal lesion patient, or animals, rats that is, were doing much more poorly on a procedural learning task, made me think about the concept of the interaction between procedural and explicit types of learning, and how those may be in competition with one another. We've done some research in our lab where we actually have subjects learning an implicit task, and if we have them do a dual task, they actually learn it much faster than if they were to be doing the task on its own without doing the dual task. And what we think we're doing is we're engaging the explicit system so that this implicit procedural system can do what it's supposed to do, and that struck me. Arpi's work, the thing that kept going through my mind listening to your talk was just this whole concept of the mind-body connection. And I think that that's really, really important in terms of understanding. It's actually a concept that I use quite often clinically in talking to my Parkinson's patients about what they're dealing with and just how these things are so important. I never thought in a million years I would be asking a patient in a clinical neuropsychological evaluation if they have constipation or not. But now we understand that it's an initial symptom that you can see in Parkinson's disease. So really showing the importance of that mind-body connection. I think another thing that your work has done is really showing the importance and the need for further tools that can be used to measure the body and how the body functions and how that can definitely impact our minds and our brain. Jared, I'm really always struck by your presentations. I'm really very, very pleased. I did a lot of work with Ray Kessner when I was at the University of Utah, and he was a big proponent of this understanding of these tasks at the animal, non-human level before we understand these things at the more, human level, I guess. And clearly having a direct pathway in terms of translating those both from non-human animals to humans, and then backwards as well, too. And I think that was really, really fascinating in terms of the work that you've done. Let me see. In terms of Mark's work, actually, Mark Shuckett stole my joke. I was simply going to say that our children's children are not going to have to go to school down the road. We will just inject them with some virus, and they will be able to know everything that they need to know. But I think it was very, very important to understand, again, going back to the concepts of unlearning, and how that can be important in terms of treatments for different psychiatric conditions. I've had discussions with Walt K. in the past about how, you know, there may be this unconscious relationship that individuals with anorexia develop between aversive feelings and what they're going to eat. And how do we get them to unlearn that? Do we do that by conscious processing? Do we tap into a conscious system to do that? Or is there something that we can do at an unconscious level where that actual memory and that relationship exists? And I think that's an important potential direction that these things can go. So let me go ahead and introduce the panel. And you'll have to apologize because I don't know you guys very well. But Sam Barnes has his PhD in psychopharmacology. And he came to us in 2011 and joined the Markow Laboratory. His work is primarily in investigating how abnormal glutamate transmission may contribute to impairments in reward and cognitive processing. And then we have Andre Der-Avakian. Am I saying that correctly? Okay. And he is a founding member of the Markow Consortium for Neurobiology and Psychopharmacology. And his laboratory focuses on developing novel cross-species behavioral and neurophysiological assessments of reward, cognition, and other behaviors in rodents, and how that pertains to psychiatric conditions. And then our third panel member is Christine Smith, who comes to us from the University of Wisconsin. She worked as a postdoc fellow in Larry Squire's lab, and her work has examined emotional learning in humans using fMRI, and she has a broad interest in conscious and unconscious learning and memory. Is that accurate? Okay, so I will turn it now over to the panel.
So, okay, I guess I'll start. I think the thing that really struck me about this session, the consistent theme not only on memory and cognition, but really a quite consistent theme on cross-species translational research and the importance of applying cross-species translational methods to really sort of understanding some of these behaviors that the speakers were talking about, and not only in animals, but how they apply to symptoms of psychiatric disorders in humans. And I think one comment that really struck me in Larry's talk early is that he mentioned that it took about 20, 25 years to develop these memory impairment models in animals. And so the comment by Larry that it took 20 to 25 years to develop some of these memory impairment models in animals, and that this problem is not just due to difficulties in developing animal models, which Jared very eloquently described his development of several of these models, and he made it look easy, but it is really a difficult task. But also due to the problems with really understanding the clinical impairment to begin with. And I think we have evidence of sort of struggles even today in trying to develop tasks based on reward processing, different cognitive impairments, in trying to develop these animal models to be accurate in terms of their reflection of the human underlying condition. And so the work that goes into that is really tremendous and is really difficult. And I think it is only, we're allowed to do this because of the infrastructure I think that we have in place in this department that I think is really quite unique compared to other psychiatry departments. And of course the support, that's a sort of a common theme that others have talked about of the department and the leadership in terms of getting into these types of cross-species relationships, and also of course just the collaboration that we happen to have in this department. People typically tend to think of collaboration in terms of clinical researchers collaborating with one another, basic researchers collaborating with one another. And I think we have really a unique environment that the speakers all really highlighted nicely, that we have quite a bit of collaboration between the basic researchers and the clinical researchers, which I think is tremendously important. And moving to Mark's talk about the implanting memories in mice, I think it's probably the only talk where we didn't hear about sort of the clinical sort of aspects of this other than with Dr. Spock. But in fact, there is some very early work in optogenetics in humans that's being piloted. I think it's quite controversial, but it may not be too long before we're able to implant some memories in in our heads.
I think that really struck me about these talks, as well as all the other sessions from today and yesterday, is the fact that cognition is so important, and cognitive impairments are evident in many, many psychiatric disorders. And so it's really, really important to try and address these cognitive impairments. But unfortunately, a lot of the pharmacotherapies that we have to date don't address them. So I think if we're able to really understand the fundamental mechanisms which regulate these cognitive processes, we may be able to begin to try and identify novel therapeutic targets which can hopefully begin to ameliorate these impairments. And I think the way that we're going to be able to do this is by using some of the approaches highlighted today in today's session, and using the cross-species translational tasks to really understand the fundamental mechanisms regulating performance in rodents performing behavioral tasks, and then trying to map that into the mechanisms for humans performing the same tasks. Using some of the novel neuroscience techniques highlighted by Mark Mayford, including optogenetics and in vivo calcium imaging, we will be able to understand with cell type and circuit specificity how the brain regulates cognition and reward processes. And using these data, we'll be able to try and identify novel therapeutic targets to hopefully guide the drug discovery process. I'll pass you on to Christine.
I wanted to bring up a connection between Arpi's talk and Larry's talk that may not have been immediately apparent. So Arpi was looking at physiology, in particular eye movements. And I want to bring up what can only be described as a 15-year odyssey that Larry and I have been on, looking at eye movements, not in psychiatric patients like Arpi, but these neurological patients who have acute damage to the hippocampus. And so as far as eye movements go, there are experience-dependent eye movements. So your eye movements change as a function of experience, so they deserve to be called memory. But what type of memory is that? Is it the unconscious memory that Larry described? Is it like mirror reading, where the eyes might betray some type of form of memory that you don't know about consciously? Or is it that eye movements so for example, if you were to look at a scene for 5 seconds, you explore a novel scene, have more fixations, let's say, in that novel scene, than if the scene were familiar. So you have this kind of repetition-based novelty effect. And so is that something that's just your eyes are doing unconsciously? Or is it something as simple as you do that because you know which scenes are old and which scenes are new? And so we designed a number of experiments to address the question of whether experience-dependent eye movements depend on unconscious memory or conscious memory. And it turned out that we were asking the wrong question, because there's a lot of different types of experience-dependent eye movements. And across all of the kinds of experience-dependent eye movements that we studied, it turned out that the ones that depended on unconscious memory were the same ones where patients with hippocampal damage were intact on the behavior, and the ones that depended on conscious memory were the ones where the patients were impaired. So this fundamental distinction between conscious and unconscious memory and hippocampus independent memory and hippocampus dependent memory that Larry's been looking at for decades now is still relevant and appears to explain new behaviors that can help inform how memory is organized in neurologic patients.
Thank you. We have time for plenty of time for questions, and I think Mark's going to.
Before we do that, could you define unconscious memory for those of us who don't work in it?
So it's the idea that you are expressing behavior or performance that improves as a function of your experience, or that changes as a function of your experience, but you can't really describe the content of what you've learned. So Larry described it like when your tennis swing improves. It's hard to actually say what it is that you're doing that improves it. You don't actually have conscious access to what your body has learned. So like, for example, when people learn to ride a bike, their body learns to ride the bike. It's a motor skill. If you ask people to describe what they're doing when they are learning to ride a bike, it has no bearing on what they're actually doing. So it's kind of a disconnect in that way.
It sounds as if it is kind of related to muscle memory. Things that we do, we've learned to do it, but we kind of don't think it through.
Right. So that'd be like a motor skill or a habit, something that you do, yeah, without thinking.
Thank you.
Questions?
Jared, I want to thank you for your elegant presentations. I was wondering, in your work with seasonal changes in dopamine and tyrosine, can you distinguish photoperiodic from circannual effects? You remember the original studies in plants that they open their leaves, even you put them in a black box at a endogenous circadian time. And my understanding from, and we've talked about the, the cognitive effects on that they tend to be better in the summer and fall rather than the winter and spring, which could be more photoperiodic history, but also could be a circannual rhythm. And my understanding from Kay Jamison's work is that the peak time of depression is actually April, consistent with TSLA. It's April is the cruelest month, and October is the peak incidence of mania, which again suggests more of a circannual rhythm.
Maybe I can respond to that. It's interesting, certainly, and some of those need to be worked. So those details need to be worked out. I don't have the carte blanche answer.
Having a bit of trouble hearing you. It's got to be
Sure.
right in front.
In terms of the observations, there are people that show have shifted mania, as you mentioned, mania in October, depression in April. The vast majority of meta-analyses suggest it's a springtime for mania. And you see, actually one of the great opportunities I had was when I first came up with these ideas. I don't know where he is. I was speaking to, I actually met with John Kelso about this, and he mentioned about some patients. Oh, there you are, John. And he mentioned some patients that he had that he set his clock by when he would get a call, and he would know that patient would be entering into a manic episode because spring was around the corner. So there are variations, of course, in individuals. In terms of the mechanism, there's a lot of very nice work coming from Sam Hattar's group showing that it's actually via IPGCRs, ganglionic cell, retinal cells. And it's actually so it's blue light in particular. There's all of these blue light filters that people are hearing about. They might actually be relatively important for maintaining, not just simply as a treatment, but maintaining a rhythm of blue light. And so there's been studies where they've given patients with bipolar mania amber glasses so that there's effectively they're a blue light filter, and that helps to reduce young mania rating symptoms, mania scores within a week if they're wearing the glasses at the right period of time. So really, I think it's this blue light, and therefore it's melanopsin associated, which actually doesn't tie into the plant. So I think it's more the melanopsin, and the likelihood is it's that as opposed to the cannula that you were mentioning. But that's work that needs to be decided upon. But thanks for bringing it up.
Other questions?
So this is actually for Mark Mayford, and forgive my showing my abysmal ignorance here. But in terms of these engrams that get developed, given that we must have, what, tens of thousands, hundreds of thousands of engrams, how does that work? Do the different cells, can they do many different things, or how? Anyway.
Don't know.
I'm sorry, I don't even know where you. Oh, there you are. My engram is faulty. I can see where you're sitting. Even. But yeah. Just as a how does that work?
I should stand up. So if you'll notice in that technique, we're labeling only the active cells. Okay, so on average for a lot of brain regions, that means 5, maybe 10% of the cells. Presumably, there are changes in limited groups of cells, right? So if you remember, I said in that first, referring to that old paper where they just randomly looked at cells and found this learning-induced change, that always kind of bothered me because of the reason that you're suggesting, is that all of the parametric space, the knowledge space, would be lost and you couldn't learn anything new because you already changed it. So the presumption would be that each individual memory is sparse, and that it involves a sort of unique pattern of synaptic changes between sparsely encoded and connected neurons. So every time you learn something new, it's a slightly different pattern of synaptic changes that occur in a slightly different pattern of neurons. Now, obviously, there'll be some overlap between different ensembles because, like I said, 5–10% of neurons get activated here. But I guess it will come down to sort of over the whole brain and over the whole processing circuit, it will come down to those sort of precise changes and the sparseness of the neurons that change.
Yeah, and I assume that the molecular changes can be very minor also at the synaptic level or...
Yes, well, so these changes are specific to synapses that are on the active neurons. We haven't actually sorted out the synapse specificity yet, but presumably they're synapses from the active ensemble of cortical inputs to the active ensemble of amygdala neurons. So that really starts to limit the nature of the changes.
This might be strange, but actually I wanted to ask the panel, since it was raised in terms of implicit versus explicit learning or unconscious memory versus conscious memory, we've all had discussions about the types of testing you can do in training, you can do in rodents as well as in humans. For example, Andre, you're in Diego Pizzagalli's task about reward learning, or the change between probabilistic learning and a reversal learning task versus serial reversal learning, etc., and how it moves from frontal to striatal functioning, after conditioned learning. Vince raised the point that it is difficult to do this and it is difficult to differentiate them, but you guys have experience in some of this work. Do you mind mentioning some of that just briefly? Especially in context, for example, I was thinking about the fact that patients with schizophrenia don't have a problem with this probabilist, this reward bias learning task that you've got, whereas people with depression do, in terms of clinical context.
Right. So yeah, we certainly see differentiation in certain tasks. I mean, as you identified as well, between NRP as well. Sure. So, you and RP identified differentiation between different sort of clinical, bipolar versus schizophrenia. We see differentiation between people with depression and schizophrenia in terms of certain cognitive tasks. I mean, I think one of the issues that you also kind of touched on, I think is important, that we need to consider between rodents and humans is that in these animals, we train them up to perform these tasks, to be able to test them, right? We don't just put an animal in the box and say, do the test, because they wouldn't know what to do. Whereas with humans, we do that, right? We have them come in, we say, do this test. And we give them, maybe a little bit of verbal instruction, but we don't really know exactly sort of the impact of that. How much is that sort of repeated training of an animal to be able to even perform the task, impacting their performance on the task relative to a human who basically just comes into it blind. And so, but there's certainly things we can do to look at that, and I know sort of you've looked at some of these things as well, looking at sort of early training versus late training. Some of the things that also you were discussing in terms of incorporating electrophysiology into some of these tasks and looking to see, as an animal is learning the task, do we see similar neurophysiological changes compared to human, let's say on the first few trials and when they're actually performing the testing. So I think there are different ways to sort of address these, but I think it's complicated.
Just one quick question for Mark. I'm sorry. Yeah. One quick question for Mark, and it actually is a follow-up kind of on Igor's. If you show somebody a bottle of Chardonnay and a bottle of Cabernet versus a chair, you've got categories of wine, and you could even throw in a bottle of vodka. All of them are alcoholic beverages. What happens to the pattern of the way the neurons light up, when they're similar category versus a totally different category?
Would that depend on the vintage of the wine?
So, that would be sort of a human-specific question where we don't have this You can certainly do it in animals, and probably the best way to look at those things is with calcium imaging rather than our labeling. Although I should say, okay, so for us, we're asking, does the animal distinguish this box from that box? They have different olfactory and different cues. And when we look at sort of how the neurons are reactivated, it's not as high as we would have expected. So it will be chance, or sometimes, in some areas actually it's below chance significantly between, say, A and B, where you're distinguishing. And when it's the same environment, it's on the order of 25 to 40% overlap. And you kind of see that similar kind of numbers with calcium imaging as well. So, I mean, one of the questions that kind of fascinates me at the moment is, if you controlled your sensory experience precisely as well as you could, just try to give exactly the same experience, how similar is the brain activity at the cellular level between those two experiences? It's just a number between one absolute correlation to every action potential to -1. And without getting into it, actually theories of how you think about memory kind of depend on how close that number is to 1, at least in certain areas, versus how close it is to more of a statistical recognition, which a lot of computational people think that is the way it works. So, I don't know, you recognize a bottle of wine, let's say there are 1,000 neurons dedicated to wine versus scotch, and anytime 200 of those are active, you have a recognition event. But it can be any 200. It's a little bit inconsistent actually with learning, which is more of a hardwired thing that says, the first time I see a bottle of this kind of wine, and if I associate it with something, it's got to be those same neurons. Those are the ones that are going to be changing based on all the theories of activity-dependent plasticity. I don't know the answer to that. I know how to pose what I think are interesting questions about it, and you get at a good point.
Good. So as you've heard, when I began in this department as a kid, there were maybe, making a wild guess, 10 people here who were doing research. And we had clinicians, and both, but of the research group, it was relatively small. And now we have hundreds. And so we are incredibly collegial. We do crosstalk, and we do translational research. But boy, we don't have enough time to have these kinds of discussions.